Hello and welcome to the next A-level video from the conscientious biologist Ben Gallagher. This is the fifth one in the nervous system playlist, and we're looking at synaptic transmission, which is the detailed mechanism by which communication can be jumped from one neuron to another. As always, I'll ask that you please do subscribe to my YouTube channel if you haven't done so already, and don't forget to have a look at my Instagram and Facebook infographics for lots of extra information that you can use in any synoptic essays and to round out your biological knowledge. So let's really quickly remind ourselves of what a synapse is. So if we're putting the whole nervous pathway back up on screen, we start with a sensory neuron. There's going to be a stimulus that hits a receptor. That's going to generate an action potential in the receptor, which you should have seen in the previous video. That might be a rod cell or a cone cell or a Pacinian corpuscle or whatever. That's going to send our signal along the sensory neuron, where it then needs to jump the gap onto a relay neuron in your coordinator. The signal is going to move through there, where it needs to jump another gap onto your motor neuron, so it can move again. And that movement, remember, by saltatory conduction, jumping through the nodes of Ranvier to get to the effector at the end, because that effector is going to give you your response. Now, when we're talking about synapses, we're specifically talking about these little gaps between the neurons. How does an electrical signal cross that gap? And it doesn't cross it in an electrical form, it crosses it in a chemical form. So that's what we're going to look at through this video. So the mechanism behind that. We can go reasonably fast because I don't think there's anything too difficult here. And in the next slide, I'm going to give you a summary, a written summary of how it all works. But let's do this one to make sure you understand it. So in the presynaptic neuron and postsynaptic neuron, we refer to them as that so it can apply to either synapse. In the first synapse, the presynaptic neuron would be the sensory neuron, postsynaptic neuron would be that would be the sensory neuron, this would be the relay neuron. In the second synapse, that would be the relay neuron, and this would be the motor neuron. So we just refer to them as pre and post synaptic. The gap between them isn't the synapse, it's the synaptic cleft. The synapse is the entire thing that's on the screen. The end of one, the gap, and the start of the other. The gap is just the synaptic cleft. Remember that, I see it every year, people's labeling the gap as the synapse. The synaptic cleft is the gap. Now what happens, we've got a resting potential in the presynaptic neuron, and you've got all these little vesicles, and those vesicles are full of something called neurotransmitter. Now link this back to your cell ultrastructure lessons. Vesicles are just little bubbles of membrane. The neurotransmitter in them will be a protein. So those vesicles will have come from the Golgi apparatus, where those proteins will have folded, they'll have budded off from the Golgi apparatus and floated down to the end of the neuron. The proteins will do their job because they folded into a specific shape, and shape determines function. Always link everything back to proteins, because remember, proteins do everything. Now in that presynaptic neuron, you're going to have diffusion of sodium from the previous node of Ranvier, same as we talked about with all the other saltatory conduction. So we did receptors to see how it's generated, saltatory conduction to see how it travelled down. We're now at the end point of a neuron. That's going to raise the resting potential to the threshold potential of -55, which is going to affect the sodium voltage gated channels to cause this influx of sodium, which is going to raise the cytoplasm to +40 millivolts. That action potential in this terminal end of the presynaptic neuron is going to trigger the opening of calcium voltage gated channels. Calcium is then going to move into the cytoplasm, bind to those vesicles, and cause them to bind to the membrane. Now that is called exocytosis. When the vesicles remember they're made of membrane, the cell membrane's made of membrane. So when they touch the cell membrane, they just open up, become part of the cell membrane, and release their proteins into the synaptic cleft. That's called exocytosis. Exo for exiting, cyto for cell, cis because it's a process. Exocytosis. Now once they're released, those neurotransmitters are going to move across the synaptic cleft by diffusion, because they were at high concentration by the presynaptic neuron. So they're going to diffuse across to the postsynaptic membrane closest to me. On the postsynaptic membrane, you've got protein receptors that will have a complementary shape to the neurotransmitter, just like enzymes have with their active site, but these are receptor proteins. But it always comes back to shape. They're complementary in shape to the neurotransmitter that's going to lock into them. Now, when that neurotransmitter locks into those protein receptors will open chemically gated sodium channels. That will allow sodium to move into the postsynaptic neuron only enough to raise it to the threshold potential, because once you've got the threshold potential, that opens the sodium voltage gated channels. Sodium floods in because it's at the concentration gradient set up by resting potential. So sodium moves in, raises it to plus 40. We've got our new action potential, and those sodiums can drift off down to the next node of Ranvier. So we've got the whole process there, the whole thing that happens in a synapse. Hopefully that's not massively complex, though there's lots of little stages to remember. So I suggest you take a screenshot of this, and then we're going to go to a full explanation. Okay, let's move on to the explanation. So this is how you could describe it if you're asked in the exam. I've put the diagram back up there so you can refer back to it. But we're going to start off with point number 1, the first thing that's happening here. And you'll note I put a 1 on the diagram to link it up to what I'm going to describe. So the sodium ions arrive from the previous node of Ranvier to raise the resting potential to a threshold potential of minus 55. At 2, that opens the voltage gated sodium channels, allowing an influx of sodium to create a new action potential of plus 40. That action potential is going to open voltage gated calcium channels. So you get this influx of calcium ions which bind to the vesicles and cause exocytosis, there's that key word there, through the presynaptic membrane, releasing neurotransmitter into the synaptic cleft. No, into the cleft, not into the synapse. Synapse is the whole thing. 4, we've got that diffusion across the cleft along the concentration gradient. 5 is what happens with these receptors, binds to the complementary, such an important word, complementary protein receptors on the postsynaptic membrane, triggering an influx of sodium ions to raise the cytoplasm to a threshold potential, that's in the postsynaptic neuron, so that the voltage gated sodium channels open to allow an influx of sodium to create a new action potential of plus 40, thus leading to a new impulse in the postsynaptic membrane. And of course, that's then just going to carry on. Now, one extra point here, so I've even put it in a different color to make sure it's separate. And you'll notice on the diagram, I've put a different color 7, because this is a recycling part now. It's additional to the signal being passed on. Hopefully you can see that points 1 to 6 is how the signal traveled along. This is then what happens as like an efficient bit of evolution. So rather than continually make new neurotransmitter, an enzyme on the postsynaptic membrane breaks it down. So if the neurotransmitter comes across, binds to the receptor, does its job, a separate protein, an enzyme, can break that neurotransmitter down. You've now got products that are in higher concentration at the postsynaptic membrane. That means they can diffuse back across, endocytose, which is basically, if there's the membrane of the presynaptic neuron, here's your products of your broken down neurotransmitter. They push, the bit of membrane here kind of swallows them, takes them back in as a new vesicle, and then can just join them back together to be reused. It's literally, they diffuse across, do their job, broken down, diffuse back, put back together. It's this continual recycling of that neurotransmitter. Otherwise, synapses would have to be making so much protein all the time, they'd be so hungry for amino acids that the system wouldn't be particularly efficient. So take a screenshot of this. This is so important for full description of the synaptic events. Really quickly, we just need to look with synapses at the difference between excitatory and inhibitory synapses. What that means is that some synapses excite a nerve impulse, cause a nerve impulse, want to send a signal to the brain. Others can inhibit the signal and stop a signal getting to the brain. This is if you're trying to ignore excessive stimuli in the body, if you're being overstimulated and you need to shut some things down. Or there's just some that want to tell you that's too much information, don't respond to that because we're processing other stuff. So it's sometimes useful to be able to shut signals up or allow them through. Now, excitatory synapses are exactly what we've been studying, where you get the vesicles excitosing, moving across the neurotransmitter, linking to the proteins, allowing the sodium in, raising the postsynaptic neuron to plus minus 55, so that you can have a full action potential and it can move down. So all that we talked about in the previous slides are excitatory synapses. Now, the key neurotransmitter for that, or one of the key neurotransmitters, is called acetylcholine. All that means is that it's a neurotransmitter that should cause a threshold potential. So like we saw on this diagram, like we've been talking about, it moves across, binds to the receptors, allows an influx of sodium to take it up to minus 55, because as long as you can hit that threshold, you'll have a new signal. Now, inhibitory synapses decrease the chance of an action potential in the postsynaptic membrane. They're decreasing the chance of you being able to form an action potential by making it harder to get to threshold potential. Now, the way in which they do that, if you look at the diagram, is they don't cause the influx of sodium, they cause the influx of chloride ions. Now, if you look at a chloride ion, it's negative. So you're not bringing a positive in. Positive ions coming in, positive sodiums, can raise minus 70 up to minus 55. But now, where it was at minus 70, you're bringing in negatives. Well, if you're bringing in negatives, that could drop it down to anything. It might go as low as minus 90. If you're making the postsynaptic neuron down to minus 90 or greater than that, you're going to make it even harder to come from that minus 90 all the way back up to minus 55. You've given it much more of a job. If it's harder to get to the threshold potential, then you've effectively inhibited this synapse from sending a signal on to the next neuron and onwards to the coordinator. Now, one of the key inhibitory neurotransmitters that we have is something called GABA. So GABA is an inhibitory neurotransmitter, is one of the absolute favourites of examiners to put into your questions. They'll say, here's a synapse using GABA. Explain why this inhibits the signal. It's just because it causes chloride ions to come in instead of sodium. They're negative, so they make the resting potential even more negative. So it's got further to go to get to the threshold. It's pretty straightforward to understand, but take a screenshot of this if you need to. Very last thing, and I've pulled this straight from one of my GCSE videos, just talking about the reflex arc and talking about why neurons are so long. Why can't we have lots of smaller ones joined by synapses? We've just gone through how synapses work. Looks pretty efficient. You can control them. Brilliant. Why not, instead of having, a 6-foot long neuron that goes from my big toe up to my brain, why can't I have loads of littler ones? That would be far more efficient, but it's not. And the reason being, it's speed. Electrical impulse travel incredibly quickly along a neuron, but diffusion is slow, and synapses rely on diffusion of neurotransmitters. Even though the synaptic cleft is really small, the time it takes for the neurotransmitter to diffuse across that synaptic cleft is significant. And if you had lots of short neurons all joined by synapses, the total of all that extra time it would take would ridiculously slow down all of your reflexes and responses. And by the time you'd realized something was flying towards your head and responded to it, too late, it would have hit you. Our brains would just process way too slowly. So we need long neurons to minimize the number of slow synapses. Why do you need synapses at all, though? It's because you want the signals to be able to link and be processed and coordinated by relay neurons. If it was just one, it would just be one closed loop where you couldn't change what was going to happen, you couldn't process the data. So you need to have minimal number of effective neurons with very few slowing synapses. So the diagrams that they always show in the exams is a reflex arc. The reflex arc will look like this in your exam paper, where you've got the spine, so the bone surrounding the spinal cord, which contains loads of the neurons. That will be attached to a receptor like the skin here, so there could be Pacinian corpuscles in here, linked to an effector, which could be a muscle. And if you've got something like a pin that's going to get stabbed into your skin, you need to detect it, and you've only got those 2 synapses that have to be that are needed to slow down the signal and allow coordination between different relay neurons. But it's very, very straightforward stuff. Okay, we're almost at the end of the entire nervous system section. In fact, we've covered everything on how nervous signaling works. We just now need to link it up to the final thing and to the effectors. The main effectors that are going to do things for your body will be muscles, because most responses will be movement based of some form, even if it's a movement to cause a secretion from a gland. Of course, glands themselves will need stimulation to create whatever they're going to secrete, whatever hormone, but mostly we're dealing with muscles. So that's the last video within the nervous system playlist, which is looking at skeletal muscle, the neuromuscular junction, which is basically a synapse between the neuron and the muscle cell, and how muscle fibers, which are muscle cells, how they contract. As always, please do like this video if you found it useful. It helps me know which style works best. Do subscribe to my channel, and don't forget to go and check out Facebook and Instagram for all the additional material. Thank you and good luck.